and welcome again. Today we look at marking of an independent investigation report in IB biology. These are the criteria that we will be using for marking the report, and we will be looking through the report and discussing what marks will be assigned and why. This particular walkthrough, of course, is not made by the IBO and it's not approved by the IBO. But I want to tell you, if you're looking at it, that the marks that I do assign for students in general over my years as an IB teacher in biology, in chemistry, and in environmental systems and societies, they are generally maintained by the IBO or lowered by about 2 marks from what I give. So bear that in mind, that whatever mark I do give this student here, most likely it would be supported by the IB, or it might just be lowered by 2 or maybe 3 marks. Let's have a look. The student's research question is, what is the effect of deferring salt concentrations, soil salinity, in the germination of Vigna radiata, which Vigna radiata are the mung beans, which what lots of biology students would work with. They tend to germinate very easily. Looking at the student's research question, the first thing that I have to ask is to do with the exploration criterion. And in the exploration criterion, I am going to begin by looking at the top mark band and what descriptors go into that top mark band. So let's take a look. The topic of the investigation is identified, definitely, and a relevant and focused research question is clearly described. Now, it's a research question, and I know what the student is about to do, but how focused is it? First of all, the student doesn't even tell me what salt you're using. The assumption is that salt, I imagine, is sodium chloride. That's not sufficient. And what concentrations? High, low, what specific range? That certainly could be included here. If you wanted to put something in brackets, may as well put the salt concentration in brackets. And germination of Vigna radiata, well, that's pretty straightforward. That's what you're going to measure. So some measure of focus in the question, a question that's pretty clear, but definitely not meeting this description here. So automatically I'm thinking maybe I should give this student something in this mark band, at least for that thing. But the rest of the descriptors deal with the background, the methodology, and safety considerations. So let's go into the student's report now and see what else they've written. But as we go through the report, it's good to bear in mind some other things. And already I'm bearing in mind one thing here. The student's naming of this seed that they're using, the plant, Vigna radiata, it's good that it's italicized and the genus name is capitalized, but the species name, that should be beginning with a lowercase letter. So therefore, communication, scientific protocol is not being followed. And that needs for me to take, that means I need to take a mental note of that. And if I go down here to communication, it says that to be in the 3 or to get the 4 maximum for this band, so to be in this area here, the presentation of the investigation is clear. Any errors do not hamper understanding of the focus process and outcomes. That's true, it's not hampering my understanding, but the use of subject-specific terminology and conventions is appropriate and correct. So, incorrect subject convention, because in biology, you're not naming the species with a lowercase letter. That is something that immediately is going to push the student out of this particular descriptor and start saying that, okay, if it's a good report and it's pretty much got a lot of the other things, then it can get 3 for communication. But I haven't gone in to look at the rest of the components as yet. That's what I'm about to do now. So going in and looking at what the student did, a table of contents, not a requirement really for an independent investigation. And also a full cover page like this is not a requirement. IB does not specifically request that students follow MLA or APA or any particular citation format. And they do provide in biology a limit for the IA of 12 pages. So therefore, there's nothing wrong with beginning with just your research question, your heading, and getting straight into the business. Again, the student starts off with Vigna radiata, this time not even italicizing the species name. So again, that error is repeated. You don't penalize the student multiple times for the same error, but you take a note of it. So I'm going to read through this document here. Vigna radiata, more commonly known as mung beans, is a plant species from the legume family. Relevant information: it is widely grown and consumed as sprouts or dry beans and therefore makes up a large portion of people's diets. I'm wondering, where did that information come from? Where is the citation? Where is the in-text citation to credit where that information is coming from? So again, that is something that is lacking in the communication area. And immediately I think I've seen enough to start feeling that this student is definitely not going to be a 4 for communication, but a 3 maximum, because 2 areas of communication have already concerned me: the species name and of course the lack of the in-text citation. There are a number of factors and conditions that affect the germination rates of seeds, and going on now to talk about all of those factors here, like sunlight, which doesn't affect all seeds, by the way. Temperature does, soil and fertilization, oxygen, of course, an important factor. Placing and planting and spacing. Mung bean seeds germinate best when planted 1 to 2 inches. Again, another issue here. The metric system is what we are looking for in scientific reports, not to be talking about 1 to 2 inches, that imperial system which is not accepted in scientific writing. So again, another concern about communication. But having said all of that, the report is pretty much easy to follow, what the student's done. And that is going to keep me looking at a 3 out of 4 so far for communication. Soil salinity, going on to talk about that. An image here which is again not cited, where that image came from, who it belongs to. Citation issues being repeated. Student going on to give some relevant background information on the topic. So I can go back to communication. The report is relevant. It doesn't go overboard with that information to date so far. It's got some structure for sure, and it's looking like a 3 out of 4 for communication. Okay, let's look at the number 1 at this top criterion here: personal engagement. The justification given for choosing the research question of a topic under investigation demonstrates personal significance, interest, or curiosity. So far, I haven't seen any great reason for that. There is evidence of personal input and initiative in designing, implementing, or presenting the investigation. I think there isn't really a whole lot there for me to justify 2. So I'm thinking by default, you get 1. Basically, you're going to get 0 for this if you don't really do a report of any substantial length. So this criterion here, personal engagement, really, it's about a 1-2. If you try your best to write a report and it doesn't show a lot of initiative and curiosity, then you're going to score 1. This student is going to score 1 for personal engagement, because the report itself is quite simple. It's not showing any great initiative. The student now goes on, though, to describe the method of the investigation, a very, very important part of the IA write-up, and it's assessed mainly, of course, under this exploration section, where we've already identified that this topic is not that well focused. The background information provided by the student is somewhat appropriate. There could have been other things provided there that's more specific to the seed and you know, to the topic of soil salinization. The method of the investigation, though, is what we are now concerned about. How good is the method? The methodology of the investigation is highly appropriate to address the research question, because it takes into account all or nearly all of the significant factors that may influence the relevance reliability, and sufficiency of the data. So you're looking really for a clearly outlined experiment telling you how to go about doing things, to be manipulating and setting up your salt concentrations, which the salt here, of course, is sodium chloride for the student. Some stoichiometry is given as to how the salt solution would be prepared, and some information about cups and molarities given. Again, this is not the metric unit of molarity. M, capital M, it's moles dm to the negative 3, or moles per dm cube is what you should write. So again, another metric unit, less significant than putting inches, but still metric unit not used properly. Some information about how to set up, but certainly not all of that information, and a lot of information missing on controlled variables. And then the student goes out of this section. Where would I put the mark for this particular experiment or this write-up so far? It's definitely going to be in the 3 to 4 band. But did I see anything to do with health and safety considerations at all? There's no mention whatsoever in the write-up here about health, safety, and environmental considerations for this experiment. And that is also referred to here, safety and ethical and environmental issues. No mention of that. So where the student was going to score 3, now I'm thinking that the student should score 2. But pushing this into the 2 mark band could be a little bit harsh. And that is the nature of scoring an IA by mark band. Sure, an entire thing here is missing, but the experiment itself is not the most hazardous thing in the world, but it would have been good if the student had shown some kind of awareness of that, gave some background information, gave a good, pretty good research question, though not fully focused, gave some details of the methodology, gave a lot of things here, and missed this one. So I'm torn between giving it a 2 or a 3, but I'm going to go as easy as possible. And I'm happy to say that I'm confident that this is a 3 out of 6 for exploration, a 1 out of 2 for personal engagement, and a 3 out of 6 for exploration. Let's look at the analysis section now. So the student has put down some raw data in a table. That's good, a nicely organized table, but look at the significant figures here. You've got 3 significant figures for the molarity, which means you are looking at this last digit here being the uncertainty, and you would want to have plus or minus half of this previous digit, or the equivalent of 1 increment here, meaning to say there are 2 uncertainties that would be consistent with this raw data. 0.001, student has 0.0005, which means they're finding half of this last digit, which is not in keeping with how you get the uncertainty. It could be the last increment that you take, that would be accepted. So 0.001 would be accepted, or half of this 100th place, which would be 0.005. So 1 0 too much here, and there's not consistency with the uncertainty and the number of significant figures shown in the experimental raw data. And here too there's not much consistency, because if you're going to put 0.05 centimeters, there's 1 0 too much here, because that 0.05 is plus or minus half of the 10th digit, which means the estimated digit is the one right after that, and you're not allowed to have a third significant figure or a third 0 here. So that is not correct. The use of significant figures, not consistent with the stated uncertainty. That issue continues to go on in the report here. The student giving 5 trials, fair number of trials, and lots of 1, 2, 3, 4, 5 data points on the x-axis for the independent variable, so that too is good, and 5 trials. So a substantial amount of data is collected. appreciation of uncertainty is there, even though it's not consistent with the data and the stated uncertainty. The idea that scientific data does have an uncertainty associated with it, that's shown by the student. And some sample calculation is given as to how to get the mean, uncertainty, and doing all the calculations, and then presenting the data as this here in this graph, with an error bar that's not stated like what that error bar means. Is it based on a range? Is it based on a standard deviation, which wouldn't be valid with just 5 trials? Mean uncertainty in the mean shoot length, I take it that could be the error bar, but it's not stated what the error bar is representing. So it kind of like kills the point of including it if you don't state why you include it. A photo of the experiment here, not very useful really to just throw a picture in unless you explain what it shows, and it's good quality. So the student goes on to analyze the results of why certain salinities grew certain lengths. Obviously the data here shows that significant increases in salinity like 0.225 moles per dm cube and 0.30 moles per dm cube lead to little or no growth in the seed. And the control, of course, grew the most. So the student has shown that you've got a weak salinity, can be tolerated to some extent by Vigna radiata. But let's look back now. Before we go on to the analysis, let's look at the student's write-up of and presentation of the raw data. This entire section here, and then the explanation as to why those results came about. Okay, so what you have is the report includes relevant but incomplete quantitative and qualitative raw data. Well, not very much qualitative at all, which is there really no meaningful pictures of how the plants grew in this salt solution. There is some quantitative data that is relevant. It is appropriate. It's fairly sufficient for this investigation with 5 data points and 5 replicates. The report does show some understanding of uncertainty, though it's not totally correct. The process data is interpreted so that a broadly valid but incomplete or limited conclusion to the research question can be deduced. All of that applies. And the question, of course, is, would this be a 3 or a 4? And in my opinion, this has to be a 3 because it's a fairly simple investigation. The student really doesn't explain the purpose of the error bar, really. There's quite a bit that says it belongs in here, but it doesn't deserve a 4. So a 3 for this as well, a 1 to start with, a 3 for exploration, a 3 for analysis. Moving down the report. Student goes on to say quite a bit in this section. Gives some meaningful photos here, which I missed to start, which I should have gone to. But now there's some photos that are somewhat meaningful, although they can't really show the growth too much. And there is some qualitative data. There is some qualitative data here now that could make me rethink what I'm going to give as the grade. And all of this actually here is about an awareness of the strengths and weaknesses of the experiment, and to some extent and the interpretation of the data, which is part of what you have to do here. The process data is correctly interpreted so that a completely valid and detailed conclusion. So there is some measure of this happening here in this section, albeit limited, and the report shows some evidence of this and appropriate and sufficient data processing. But again, I am tending to put this more in the 3 bracket instead of the 4. All right. So moving down to the evaluation, the conclusion and the evaluation. This part of the report, we are looking for The top mark band again as the starting point. A detailed conclusion is given. It's described and it's justified. Strengths and weaknesses of the investigation are identified. Realistic and relevant suggestions for improvement and extension of the investigation. So these are all things that I'm going to look for here in this conclusion. And what the student is about to conclude here, if you have a look at this, is that salt solution, if it's too high, it's going to affect the germination of Vigna radiata. And if it's lower, it won't affect it. Pretty much a straightforward thing. Uncertainty, simple things, number of trials, uncertainty with the measuring equipment, dormant seeds, other variables, human error, and you can read all of those. And immediately what it would suggest again to me is, again, because of the simplicity of the experiment, it becomes difficult to get into the top mark band. But again, a conclusion is described which makes some relevant comparison to the accepted scientific context. No, there was no attempt to do that really. Strengths and weaknesses were identified though. Some realistic suggestions for improvement. Let's go back to the student's report and see if there were any suggestions for improvement. Yes, this can be because of salt falling on, therefore, while performing such tasks, close attention must be paid. Artificial light coming from the lamp. So lots of issues identified, simple ones, and some recommendations made. So the student has identified some strengths and weaknesses, albeit simple ones, and made suggestions for improvement. Conclusion is described, and it makes reference to the research question. Again, can't be more than a 3, and it's definitely in this 3 to 4 mark band. Again, the simplicity of this activity. Just putting your mung beans in salt kind of like always is going to end up with things leaning towards a 3 instead of a 4. Good communication throughout. It wasn't a difficult report to understand, and there is a bibliography here. Some sources recognized at the end, even though there weren't any in-text citations, which is good to have. Communication generally was it was easy to follow, and I think it deserves a 3 out of 4 for communication. So we are going to give this 3 out of 4 here, 3 out of 6 here, 3 out of 6 here, 3 out of 6 here, and 1 out of 2 here for personal engagement, giving this report a total of 1 plus 9 plus 3, 13 out of 24. And 13 out of 24 is certainly not a bad score at all. Students might be thinking, oh, that's really a poor score to get in the IA. But what, of course, it shows is that the student has pretty much shown a good understanding of what goes into the writing up of a lab report for the independent investigation. Of course, there's a lot of room for improvement, and by following a couple of simple tips, this score could be improved to as much as about 16 or 17, but it's kind of like doomed to stay around there because of the nature of the topic. You would have to possibly do more than one seed. You would have to do some research into why is it that you want to investigate this for Vigna radiata with sodium chloride and with that range of concentrations. And if there's some justification in the literature, which means in some research paper that you've read that has led you to say this is what I want to investigate, or maybe I want to investigate it with more than one seed, making your question a little bit more rigorous and a little bit more original, of course. That immediately can change all of those 3s into 4s, giving some kind of personal reason why you want to investigate this thing, and being a little bit more involved and engaged in terms of like being creative. You can show that, collecting extra data, investigating more than one seed, making some kind of creative and innovative design for your experiment. All of those can show personal engagement and can give you a 2 here. Here, a simple thing the student could have done was to show safety and ethical considerations, and that would have given a good chance of a 4. Also, be aware of all the controlled variables during this part of the write-up also, it would have been good to use a couple photos to explain exactly how things would be laid out, to control variables and to measure the dependent variable and to manipulate the independent variable. And that could easily push the student up to a comfortable 4 here. Here, of course, there was the error bars weren't explained why you have them. There was a problem with the uncertainty, and that could have, if those were fixed, this could have been a 4 here. And generally, of course, the evaluation was what you would call quite superficial and simple, and that is really a product of how simple the experiment was. If the experiment was simple, there's not going to be much chance to get up into the top mark band for this. But the student could do themselves a favor by making some reference to other research, making suggestions for what I might want to do next, how I might want to extend this investigation, and those things can also push this up to a 4. And then overall, good communication, though. It would have been hard to get this to a 4, but if you didn't have the error with the naming here to start with, the use of imperial units instead of metric units, and the uncertainty issue, which was penalized in another section. So just a couple of communication points to improve on could have possibly made this into a 4. And then suddenly you would have 4 plus 4, 8, plus 4, 12, plus 4, 16, plus 2, 18. 4 4s plus a 2, 18. So you would have, on the one hand, with a lot of positive marking and changes, really rigorous changes, you can get this IA up to an 18. And I'm giving it right now, as it is, a score of 13. Likely that if the student pays attention to a few things, that this generally could be given a score of 15 or 16 with a couple of changes. So should you do this particular thing for your independent investigation? No, you shouldn't do it. It's just here as an exemplar to give you an idea of what you need to do. And of course, the whole point of IB giving you this independent investigation is so that you can find something that is meaningful to you from your time in the IB diploma and choose that. Usually at the end of 1 year of study, after exposed to lots of topics, you want to get to something that you can do to show your creativity, your understanding of the scientific method and how to write up a lab report in science, biology, or chemistry, or environmental systems and societies, which is not a group 4 subject and it's got a slightly different mark scheme to this, but quite similar in ESS, and of course the same criteria in physics. And the idea is not to go use this as your IA if you're watching this on YouTube, or to go on some forum and see what other people might have done, or to even source someone who is willing to write an IA for you. All of these things are unethical and illegal, and it's not the intent. The intent is that you would spend your time reflecting as your teacher goes through each topic in the syllabus. Something would come to your mind as to, maybe I want to investigate this, or I want to investigate that. And when the time comes and the teacher says, go ahead and do your IA, this is what you would choose as your topic, the one that you would have had in mind as your topic for your independent investigation in biology. And immediately, you won't have to be lying or kind of having some kind of contrived personal engagement, because it would become natural. And of course, you would be very involved in searching for an answer, researching it, and the opportunity to score in the 5 to 6 mark band would kind of appear to you organically, provided that you have the right guidance from your teacher, which you're going to be getting from me, of course, and you would be able to score up there in the 7 mark band, which is going above 19 into the area around 20, 21, 22 for IB biology.